micro nano education for everyone.
Our next presentation is going to be by Dr. Sandra Porter, who is a biotechnology, works for Innovate Bio. I think she's a co-PI for the Innovate Bio project. And she's going to be talking about an antibody hackathon that I'm hoping that some of the students in this group will participate in. So thank you, Sandra, and looking forward to this presentation.
Thanks, Jared. I want to say, if you guys want to learn about computational thinking and get some practice with computational work in kind of a great setting, working with teams, I really can't not stress the opportunities that are offered through hackathons, because I was in my first one this summer, and my husband has been in some, and they're a great opportunity to work with others and teams to work on computational problems or other kinds of problems. And yeah, they're a lot of fun. And so I'm going to talk about one that I hope that you're interested in participating in, which is going to be with antibody engineering. So first, I want to share my screen and show you a little movie. It's really short, but it kind of gets to the point. And I put the URL for the movie here. And this is about the project that we have with the National Science Foundation. And I know some of you guys have done some great work with antibodies. I looked at the posters. And these are members of our advisory board. We have all kinds of people from different companies that work with antibodies or cell engineering that are participating and helping us out with developing projects and answering questions from students. Okay, well, so much for the movie. So what the whole project is about is that antibodies, you may have gotten the idea from the video, antibodies are, I would have to say, one of the most important products that are made through biotechnology. In fact, the 100th antibody was approved for clinical use for treating diseases a year ago in March. And there have already been 8 antibodies that have been approved by the FDA for treating COVID-19. So antibodies are like a really, really important tool. They're used in a wide variety of things, not just as drugs, but also as they're used for diagnostics. They're used, any kind of tests. A lot of antibody tests for COVID-19. There's antibodies are used as reagents in any kind of field. If you ever see cells that are stained and look lovely, right? Fluorescent cell pictures, those are stained with antibodies that are binding to specific proteins in those cells. Even people in plant biology use antibodies. Okay, so antibodies are proteins, and we can make them. We're not even limited to just animals anymore. But working with antibodies is great, because if you look at our page here from Biotech Careers, you can see worldwide, we have made this database, and we have 484 employers in over 1,000 locations in the world. If we look at the United States, we have, actually, I forget, it varies from day to day. We have 355, and you can see they're like all over the country. All kinds of people make antibodies. There are even people that make antibodies to things like snake bite and stuff that just, I don't know, I guess they buy horses or cows and inject them with snake toxin and drain their serum, and then they sell that. They sell those products. So people make antibodies to lots of things, and they can make them in vitro now. We can make them in yeast. We can make them in fungi. We can make them in algae. So it's really, really important technology. But so how do you get the skills to working with antibodies? Some people are getting skills to work with antibodies in their classes, but we think that there could also be a lot of research projects around antibodies, and having worked with research projects with antibodies, this would be a great thing for you when you're looking for a job, or if you want to go on to graduate school, or the kinds of things that I know Jared's students have done with antibodies, which are neat. So how do you get that practice? Well, we thought it would be a great thing for faculty to have different kinds of research projects that they could put into their classes that are based around antibodies or antibody engineering. And we take a broad view of what you might do with antibodies. But how do we get those projects together? How do we get them started? So we thought a great way to do that would be to do this through a hackathon. And now you may have heard about hackathons being really competitive, hardcore competitions. And sometimes that's true. But we're actually working on a hackathon where people can work together in teams. We view hackathons as a triangle, all right? And part of the triangle is, one end of the triangle is community, one end of the triangle is what you're going to learn, and the other end of the triangle is what we're going to build and what we're going to make. And the things we're going to make are these kinds of research projects. Now, we're starting out with research projects that were suggested by our advisory board. One of them is a project to work on a tool for molecular modeling. It's a free tool called IC in 3D that is made by the National Center for Biotechnology Information at the NIH, the National Library of Medicine. And you can come here to our site or go to their site and give it a try. It's really fun. We want to make an educational version of it. We'll also be using IC in 3D to improve it and add different features. It's really kind of a powerful tool. People use it for all kinds of things, for doing some of the machine learning that we were hearing, with looking at detecting variants of concern with SARS-CoV-2 variants that might be escaping antibodies. Which brings me to the next project. This is one I'm going to be working on doing in my class in a couple of weeks with looking at anti-SARS antibodies. We'll look at some of the commercial ones, and we're going to use IC in 3D and do some modeling to try to figure out how well the commercial antibodies might do against some of the variants that are appearing with SARS-CoV-2. So that's a type of research project. Another research project is going to be around the immune epitope database, which is a database out of Scripps Institute in La Jolla, and they are looking at identifying parts of proteins that are recognized by antibodies and T cells, and that information is used for making vaccines and making drugs and all kinds of things. But we're not just doing computational projects. We are also working on developing lab projects. So our advisory board thought a really good project would be for students to use modeling to look at the interactions between an antibody and antigen, and then figure out how to break that. So we can design primers, and we can make new genes, and we can figure out whether we can prevent an antibody from working, and do staining to see if it's binding to something else. We also, one of our co-PIs has this, he and this guy from this company have this idea about making this low-tech antibody screening method. So you could use yeast display to try to design all kinds of new antibodies. So we think that could be kind of cool, because it could be used, if it all works out, in all kinds of institutions. So there's all kinds of projects. This is what we have for the first year. You get to sign up for one, and we'll kind of divide people into teams. But everybody will be presenting on their projects. We'll be learning about antibody structure and about antibody engineering. And then, so how will the hackathon work? It's going to be Thursday, Friday, Saturday, Sunday. We have a half day to have a kickoff and start on January 13th, and then kind of days where people check in, and then they work Friday and Saturday, and then Sunday presentations. And then people will be working on the research projects, what do you need for the research project onward? And all the work that we do, we will be putting into Cubes Hub. Some of you may have heard of something called GitHub that's used in programming, software engineering projects. Cubes Hub is a similar thing, except that you develop curriculum and then people can extend that curriculum and carry it forward and do other things. Does anybody have questions?
Is the hackathon open to new suggestions or new proposals for projects?
You know what? This year, maybe not. It depends how many people we have sign up. But
Okay.
next year, for sure, it's a 3-year grant. So,
Okay.
but if you participate this year, you can definitely suggest things for next year. There also is going to be a hackathon this summer in Madison on protein structure that's part of the International Society for, I always forget what ISMB stands for. It's like an international computational society that meets every summer. It's like the World Bioinformatics Professional Society. And they're going to have a hackathon on protein structure this summer with IC and 3D, because they're always trying to figure out how to make it better. Oh, yeah, one more thing I should show you. So if you go to our Antibody Engineers website, this is cool. You notice at the top, and this is just antibody-engineers.org. At the top, it says structures. Okay. If you go there and you click that, you can actually play with some of the structures. If you just click one of these, you will open the structure in IC and 3D. And you get to get an idea what it looks like. And this is IC and 3D, so you've got like all the tools for looking at the sequence, the structure, and the function. You can highlight things. You can change the way things are drawn. It's all free. It's pretty fun.
Is there any kind of computational limitations that we would have with laptops? Like, do you need a certain amount of RAM, or do you need like a certain like schematics to be able to run these programs?
I can run IC and 3D on my iPad.
Okay.
I don't actually know the RAM limitations, but it is, what's that?
It would just have to be modern web browser that does WebGL.
A modern web browser that does WebGL.
Okay.
I have found actually Firefox is really the best thing, because there are some features. Well, so one of the things we're working on trying to do here is actually, trying to fix some of the bugs in IC and 3D. There's one programmer who works on this at the NCBI and an intern who's working with us. And so really, if you want to play around at doing some programming and learning how to do JavaScript and UI design and have some fun, this is a good opportunity for this.
Do you have anything that would pertain to like stuff like antibody in regards to like cell manufacturing?
Well, it's not some, yeah, actually, antibodies, there is a relationship between antibodies and cell manufacturing. I imagine you may have heard of CAR T cells. Have you heard? CAR T cells are chimeric antigen receptors that are put onto T cells. Well, half of that chimeric antigen receptor is an antibody.
Uh-huh.
Okay. The part that binds to things, the part that, interacts and is specific and can recognize things, that is an antibody. And it's joined through genetic engineering to G-coupled protein receptor, I think, so that you can put these genes into T cells. The antibody makes them specific. And then when they bind to something, the G-coupled receptor turns on the T cell and it gets ready to, it activates the weapon. So it's going to kill a cancer cell or something like that.
That's good to know.
Yeah. So anyway, it's a technology that's already really important, and it's only going to be increasingly important. And I helped develop that GAP-C project, by the way, when I was at Geosvisa. It was really funny because some researchers that were working on GAP-C were going to be collaborating with Biorad. And we provided them with the LIMS, the Laboratory Information Management System. And have any of you guys used Finch TV? Yeah, we made Finch TV.
Thank you, Sandra, for the presentation. I just want to jump in quickly to ask a baseline question. If I'm a student, I'm really trying to understand how to get involved. Like, what are some skills, what are academic subjects I really should be working on and trying to get really proficient in now? And really, this is for anybody who presented today. What are some like those foundational academic skills I should be working on right now? I know somebody was suggesting learning Python. I think that would be good. Python R is also really good. I think actually getting involved in hackathons is good. Starting to, you know what else would be really good that I don't really see many people actually ever doing, is starting to try to force yourself to dig through and decode scientific papers. So you may not know this, but the NCBI they have like 38 different databases. And 2 of the databases are literature, scientific literature. And I think one of the things I had the hardest time with when I was an undergraduate and I ended up having a research project is people just handed me these papers and I had no idea what to do with them. And I didn't even understand them and I was afraid to ask. So I think actually starting to work on some of those and trying to figure out like how to read those things and how to get used to, the how things are presented is probably a good thing to do. And a whole bunch of this is free now. It didn't used to be free, but it is now. And I find that very helpful.
So my thought to add on that is I think take advantage of programs like what you guys are running. To me, that's critical. And seek mentors that you enjoy interacting with and can help you. And the other thing is one of the hardest things for students when they transition to research is connecting the fundamental knowledge they got in the classroom with what they're actually doing in research. Sometimes when you do research, you miss the forest for the tree, right? You're trying to get this code or back out of your code and running this, and you don't see the big picture. Once you start putting things together, now that takes time, but try to think about that. The stuff that you learn in school, there's a reason for it, even though it might not look like when you're taking the classes. And don't be afraid of taking the harder classes. That's okay. And maybe you don't get an A, but that's going to push you a little bit away from your comfort zone and provide you with foundational knowledge. It's very hard to do that later, right? You're an undergrad student, take the courses, try to get as much information as you can, because that you're going to build on that throughout your career.
Thank you all for that. And Dominic, I saw your hand raised.
I'd like to chime in.
One of the things I wanted to touch on was like Dr. Shark, you talked about the core academic subjects, like what actual academic subjects. So it was like what math, what science courses.
So in my mind, that's more discipline dependent. Biology will be different from my background is in physics and material science, but I would say the basic math, programming for sure. If you can take a course that walks you through the formal aspects of programming, whether it's Python or Java, right? It doesn't matter. Once you know how to think about these things, it doesn't matter what language you use. It's very easy to switch from one to the other. But just whatever your discipline is, the basic courses on that. And sometimes courses that maybe are complementary to what you're doing, right? If you're biology, maybe advanced math courses allow you to differentiate yourself from most bio people who maybe, Calc 1 is enough. And so I think you just have to do things that you enjoy and just pay attention. And these will that background, right, is just basic skills. Eventually they'll kick in, you know, and they'll be useful for your future.
Yeah. I know we're coming up on time. I want to be respectful of that. Dominic, I wanted to give you a chance to jump in.
Oh, I just want to say I run a biomed tech training program at Community College of Philadelphia. And for our purposes, I think to get the most out of the program, you want to have, like some have said, a basic computer science course, maybe an English composition course, a basic chemistry course. And then particularly for biomed, a cell and molecular biology course is pretty valuable to have a basic scientific literacy to get the most out of those experiences. That's just my 2 cents on it. That's all.
I like the English part because communication is critical, right? We don't do research for ourselves. Part of doing science is communicating science, and part of doing science, communicating to the public, but also communicating within groups and collaborators. So learning how to work together, learning how to look at the person sitting next to you and asking, hey, what are you doing? What are your thoughts? And how you can put 2 + 2 together. That's a big part of doing innovation. Often it's just saying, okay, hey, you know how to do this. I know how to do that. 2 + 2 is not 4. It's easy. Yeah, but you have to learn how to talk to people.
Okay. Yeah. Now with that, it is kind of hard, you know, when we're like, we're because of the pandemic and, you know, things like that, we're not going to be together and we're not going to be able to say, okay, partner, you know, can you help me with this? It's going to be, you know, 10 times more difficult to even interact with anybody. So, and to follow up that point on the, as far as the classes we need to take. Now, are these programs offering like the basic knowledge we might need, or like as far as, for example, somebody was presenting earlier and they were speaking on T cells, and you know, me personally, I was forced to take specific classes because of my degree. So I have a better understanding than everybody, but I know there's a lot of other people in here that don't know what a T cell is. They don't know what anybody is talking about as far as that. Now, are these programs, when we join these programs, that they're going to give some type of background, like, okay, well, a basic understanding of biology, we're going to give you this, and then we're going to move on to, you know, the more complex things? Or is it you must, you know, already have this knowledge or already have taken these courses in order to progress in this program?
I think it depends. Can I answer for that? There are biotechnology programs. There's at least 130 around the United States. They tend to be very focused on getting people into the workforce, but they will be very focused on the kinds of subjects that are important in their area and localized. So some programs might be focused on engineering plants because they're in St. Louis and that's what people do there. Some programs like in Seattle, they're very focused on cellular engineering, cells and biomanufacturing, and T cells because we engineer T cells here. So they teach you the knowledge you need to know to go out and get a job. And then oftentimes the companies will pay for you to continue your education and get a deeper knowledge if you want to move up and get a bachelor's degree or master's or something else.
I would like I agree with you completely, Sandra. And I would like to also add, I'm in the California Institute of Regenerative Medicine biotech program. And starting off, I knew very little about anything about biotech. But the thing that really helped me in reading the scientific papers was joining a journal club. Because a journal club is, it kind of, you read the papers and you discuss them with peers, so you gain a better understanding of them. And the great thing about that is in the process of learning whatever subject you're interested in, you learn how to read scientific papers in general. So you can start to read papers on your own field of expertise. And I would kind of encourage any group that starts here to start a journal club of your own for your independent projects. So, because you have to do background information on any project you do anyway, and that would be a great opportunity for those who are less familiar with scientific papers to get a familiarization with them. Because we're all at different levels coming into these projects. Some of us know more than others. and some of us, but it could be a great opportunity for everybody to have a journal club and develop kind of a common understanding of what your research is all about.
Great idea. Plus, you have to say the words out loud, and that helps.
Yeah, I didn't know how to say mesenchymal or mesenchymal, or I found out both words are accepted, but you'll never know that until you do. So
awesome. Any other questions? These are really great questions.
Yes, I sort of have a general question. I'd like to put out that, well, it's general. I'd like to apply it to either Miss Sandra or Dr. Sturgeon, as well as to you, for advice or I guess comments specifically on my situation, as with all the throughout the consecutive weeks and everything that I've been putting into consideration of what I wanted to do for the project would be a continuation, I guess, of my high school project, which was on cryonics. And I guess I would, of course, need to do further research in order to update myself, since that was about a decade ago. And I guess I'm looking for like where would the best path to be in order to continue. I'm currently computer science, engineering major, but I'm switching it to computer science, taking computer science classes. And to my understanding, where we left off with cryonics was that, and we were looking at, and there was hopes for nanotechnology to be able to overcome the main hurdle, which was when you freeze a human cell, water inside the cell crystallizes, puncturing the cell, killing it. And then, and with cryonics, there was a special crystalline formula that would, when solidifying the cells, it would make it solid, being able to preserve it. And they were looking into some, and the major hurdle was bringing them back from that point of cold preservation, in which nanotechnology would be more than likely the main solution. So I suspect I would have to look into biology, as well as the nanotechnology, as well as being able to compute or needing some type of computer precision to be able to have the nanites be able to perform this procedure, in which case, I guess in that jumble for comments or advice.
Well, really quickly, Score, thank you for your question. There's a couple of, there's a couple of suggestions in the chat. In fact, I think you're at the same university, so Dominic, do you want to jump in?
So we're at the same university. That's terrific. So I'm just first timer here today, so thanks. I'm getting to know everybody, and I don't have a lot to offer there, but we do have a biology club that we just started at CCP's campus. And I think that might be a good basis for gaining some of that knowledge and literacy to connect with other like-minded people and really be able to improve on those skills. And I'm thankful to get involved. So, and you and I can open up a dialogue too, and definitely work together moving forward. So make sure you get in contact with me.
And Sandra dropped a note. Sandra dropped a note in the chat to look for papers by Mark Roth.
I'm not that familiar with cryonics, but there is a researcher who I think he might still be at Fred Hutch. He was somebody I knew when I was a postdoc there, who discovered that you could put mice into hydrogen sulfide and they would live and preserve them that way. Is that right? I probably have this. Yeah, that's right. Yeah. So it's an alternative to freezing, but it's a preservative. And he's been doing work on using.
Actually, it's not hydrogen sulfide, I don't think.
I think it's like molecular. Yeah. Halogens. But look up Mark Roth, because he just got a MacArthur Genius Award not too long ago for this kind of work. And it's pretty neat.
Thank you.